I'm gonna record this part right here. Okay, so, all right, this is bad. I didn't mean to start my recording with this, but you guys, I know like we don't spend the lecture today just taking pictures of all the slides because I know you're not gonna look back at it. Right? All right, so please, I'm going to encourage you, take notes. Take notes. Don't depend on your phone because I know whenever I do this, I'm not going to look back at it. All right, I'm not going to look back at it. All right. Okay, so that was bad start to this. Now I know what's next. Okay, so next one, Jesus sent this one to me, but someone sent this to me last year, and I thought it was so funny because it's kind of true. It's like what we study in class versus what we see on the exam. Now, to help us be more prepared for the test on Monday, though, because I know sometimes you're like, what? Like the test was so much harder than what we do in class. It was so much worse. Now, so that you're not shocked by Monday, the progress check is going to be very similar to your test. All right? So I don't want people to be like, wait, like that was way worse. Progress check is going to be very similar, same types of questions that you'll see on the test. Okay? All right, now I think we're really going to get into it. So all of these objective slides, I am not going to read over. They are on the PowerPoint. I'm not going to waste time going over it, but you go through all the objectives to check to see if you know everything. Okay? So all these objective slides, I'm skipping them for now, but when you study, go back and review. So as a recap, the 4 macromolecules were carbs, lipids, proteins, and nucleic acids. If I ask you, you're lucky you can use your notes. You don't have to memorize anything. But for like in previous years, you would have to know all of these by heart. You should know the 4 macromolecules and the elements that are in them. As a recap, dehydration, synthesis, and hydrolysis. Dehydration, synthesis, you're building up. It involves the removal of a water molecule. And then for hydrolysis, that would be breaking down. I would make a note actually right now. This word hydrolysis on the test, hydrolyzed. Okay, so if they say this molecule is hydrolyzed, it means it's breaking down. Okay, oh my gosh. I'm trying to see the chat right now. Okay, focus. All right, so Jesus sent this one to me too. So you thank Jesus for all these. I had to look it up because I never saw this movie. But when your body breaks starch down into single molecules of glucose by consuming water molecules to separate the chain, Kowalski hydrolysis. No, it's close. All right, okay, moving on. So this table right here, you are make sure you have this memorized, the macromolecules. You should know the monomers for each one, and then you should know the elements. This element section, this is going to be, it was also on your pattern matching, slide number 2 or 3 on your pattern matching. So you review this. This should not be something that you want to waste time on your test. This was the slide. So I remember it as, to help me remember, I remember it as CHO. Oh, you can't see the text. So CHO, CHONS, and CHOMP. The exception would be for lipids. Sometimes they'll have nitrogen, sometimes they'll have phosphorus, but only for phospholipids. Okay, so the way I memorize is CHO, CHONS, CHOMP. All right, we started this unit with this question, could life exist on Mars? I gave you this slide that said atmospheric contents of modern Mars. So of all these molecules, you figured out the chemical formula for it. What I would like you to tell me in the chat right now is, based on what you know about biological molecules or macromolecules, does the Mars atmosphere provide all the necessary elements needed for life to exist? Okay, so look at these elements listed here. And then now what you know about biological molecules, which was this chart. Okay, so the elements needed. Would you say life could exist on Mars? Yes or no? Can you tell me in the chat, yes or no? All right. So what you're saying, looks like most of you are saying no. Now, that based off this, you're correct. So what is missing? What elements are missing that the atmosphere of Mars does not provide?
I mean, it has water, but it's mostly in the form of gas.
Okay. All right, yeah. So they always would make a big deal like, do planets have water? Because water is the basis of living things. Now you can think about like, yes, it's in the form of gas, but let's look. If we look at these elements, what elements are missing? So we've got hydrogen in water. So hydrogen is present in water. So it's here. But we have carbon. Yes, Aranza says sulfur. So sulfur is necessary for proteins. There's also one more element that is missing. So we got carbon. So far we have CHON. So we have C, hydrogen from water vapor, oxygen, and then we have nitrogen. Sulfur is missing, and then the other one would be phosphorus, which is needed for DNA and RNA. Yeah. So according to what's on the atmosphere of Mars, there's no way we can make DNA and RNA because there's no phosphorus available. So when they were first collecting samples, this was their first sample. Now on Earth, on ancient Earth, this was what scientists believe was on ancient Earth atmosphere, or what the atmosphere on ancient Earth was made of. One thing to notice is there is sulfur present. However, there's still no phosphorus here. Okay, but we know that life on Earth has existed. So, I mean, we are living here. So the phosphorus had to come from somewhere. And similar to Mars, what they found out is on Mars, it was actually in the geological sample. So geological would be like the soil, the rocks. So I know this picture is really blurry, but in the geological sample from Mars, scientists have figured out that it does contain silicon, iron, sulfur, and phosphorus. So in theory, the geological sample combined with the atmospheric sample, it could provide all the necessary elements for living things. So we'll see. I mean, they're still doing a lot of research, but based off the elements, it is possible. It is possible. Okay. Now, I'm going to start getting into each of the macromolecules. Okay, so we're going to go in order of the textbook. Every now and then I'll point out pages in the textbook to refer to. So if you have your textbook ready, it might be useful to like put post-its on certain tables for you to study. The first part is going to be carbohydrates. All right, some of these slides were from last week, but I'm going to go over the basics, what you should know about carbohydrates. So carbohydrates are also known as polysaccharides. If you see the word polysaccharide, you should automatically think carbohydrate. The monomer of this is monosaccharide or simple sugars, or if you see something like glucose, fructose, those would all be examples of monosaccharides. One thing that I would, hopefully you recognize is a lot of these sugars end in ose. So if you see a word that ends in ose, it's probably going to be a sugar, which is going to be part of the carbohydrate family. So I could think of sucrose, lactose, galactose, any of those words, if you see it ends in ose, it's going to be a sugar. Okay, this was part of your pattern matching. You can see that sugars could be a ring-shaped or they could be linear. All right, one thing though that tells me it's a sugar is that in these rings, it's just carbons, hydrogens, and oxygens, C, H, and O. So structural features of carbohydrates are CH2O, meaning this is the ratio of carbon, hydrogen, and oxygen. They can be ring-shaped, but also linear. This was the same slide as last week, so I think you should have this already down. Okay, moving on. So this basics of polysaccharides. Now, one thing with sugars, if it's a ring shaped and actually linear too, um, we number the carbons in this direction. So it's kind of weird. It's clockwise, but looks backwards to us. So we look at glucose. This first carbon would be carbon 1, 2, 3, 4, 5, and then this one at the top would be carbon 6. This will come into play when we talk about nucleic acids. But just so to give you an idea, though, they number carbons. So if they say carbon 1, and it goes in this like clockwise direction. Now, polysaccharides are chains of monosaccharides put together. So this image up here, you just see there's a bunch of glucose molecules all chained up. They are polymers of monosaccharides or sugars. It costs little energy to build, and it's easily reversible. And when it's reversible, it releases energy. So when I say it's reversible, it means that it could be broken down. So I can cut off basically each of these glucose sugars, or not glucose, but any of these monosaccharides. They could be broken off piece by piece. And then when this is broken off, this single monosaccharide can be used in cell respiration to make ATP. Now, for polysaccharides, the section like this, the next few slides is going to be focused on structure and function. So polysaccharides, the function can be for energy storage. And the example for this would be starch in plants. So like potatoes, carrots, those have a lot of starch in it. It's stored up energy for the plant. It can also be stored as a form of glycogen in animals. And so humans, we are considered animals. We would have this in liver and muscle. If we don't have any single glucose, molecules in our bloodstream, your body goes to your liver and your muscles, and then they break down glycogen. It can also be used as structural components. So this would be cellulose in plants. This is found in the cell wall. So this picture right here, this is a picture of a plant cell. And hopefully you remember from first year bio, but the plant cells have a cell wall, and that's what gives plant cells this rigid shape. It's also a part of chitin, which is in arthropods and fungi. So it's the what makes up the exoskeleton. That's what makes bugs crunchy if you step on it. That would be chitin. So these 4 terms, starch, glycogen, cellulose, and chitin, I want you should be familiar with their function. And then I'm going to briefly talk about their structure and how it's related to their function. This is in your chapter 3 study guide. So there's a whole section of this in your chapter 3 study guide, number 27 on your study guide has a table that goes over this. Okay, so for glycogen and starch. So starch and glycogen, you can see the simplified structures right here. Notice each one of these is a tiny little hexagon. These are all small glucose molecules chained together. So energy storage, starch in plants, and then glycogen in animals. Now for starch, there's 2 types. They're listed here. You don't really have to know, the details of that, but you should know that starch is for plants and glycogen is in animals. Cellulose, it looks very different. It looks very, they're all lined up very orderly. And it makes sense because this is part of the cell wall. It's used for support. So you want something very orderly and sturdy. So one thing about these, comparing all these molecules is how the way they look, their structure is related to their function. So for starch and glycogen, I made a little note here. It says when backup energy is needed, we go to these 2 molecules. Starch and glycogen get broken down sugar by sugar. So if you notice with these molecules, they are branched. And the way your body or plants would also break this down is they'll cut off each sugar like one by one, starting from the ends. Okay, so like the way I'm cutting it. So they'll cut it from the ends, the end of the branch. Same with the glycogen. So if you see there's multiple branches, so there's lots of places where they could start cutting sugar molecules off, monosaccharides, so that they can be used for cell respiration. The branching, you should notice the difference in branching. Which molecule has more branches, starch or glycogen? Tell me in the chat, please. Which molecule has more branches, starch or glycogen? Yes, glycogen. So if you think about it, if this is an energy storage molecule, who's going to need more energy at a faster rate? Plants or animals? Do plants need to like move around and run around? They don't, right? So for us, it makes more sense that animals have more branches because that means more sugars can get cut off. Plants, they're just they don't need to like be running away from a predator. Okay, so yes, they still have this backup energy storage, but they don't need to break off as many sugars all at once versus animals might. So that is one thing for how structure is related to function. So starch versus glycogen, the branching, that would be related to the function. So plants don't need as much quick, energy versus animals. And then if you were to compare starch versus cellulose, these are both in plants, but obviously starch, this gets broken down for energy. Cellulose, they're very, I mentioned this earlier, it's very organized in the way that they're structured because this is for strength. Okay, the next few slides I think it's similar to what's in your textbook, but the difference between starch and cellulose at the molecular level, it's a very tiny difference, but you could see for starch, all the oxygens, the bonds are in the same direction, so they're kind of like facing down. And then for cellulose, you could see that the oxygens, the bonds kind of switch back and forth. So this is just a slight difference in the type of glucose that is being linked up, but this tiny difference gives you the big difference in the structure that you see here. So just this tiny difference in the bonding pattern allows cellulose to be not digestible. So cellulose is the stuff that our bodies can't break down, versus starch, our bodies easily break it down. Okay. This last one also is, I think, similar picture in your book, but you could see in the starch, each of these glucose monomers, all the oxygens, the bonding they're all in the same direction. Glycogen, same thing. But then for cellulose, the oxygens, like, one's going up, one's going down, up, down, up, down. They switch off. Starch would be things like potatoes versus cellulose. The example that, maybe this will help you, but if you eat celery, I hate celery, but if you eat celery, I don't know if you notice, it's always like very stringy. Like when you bite into it, there's like all these like strings and fibers. That would be like kind of like cellulose. And our body doesn't break it down. It will just pass through us. Okay. There are practice questions, but I'm gonna skip them because I don't think we have time. Actually, you know what? Let's do this one. Which of the following best describes the structures of carbohydrates? Don't say it in the chat. I'm gonna put a Zoom poll. Okay, so let me launch the poll. All right, remember on a test, it's about 90 seconds per question. So this is a pretty short one. I don't think you need 90 seconds for it. Okay, that was 90 seconds. So here is your result. The majority of you picked letter B, and that is the correct answer. So they occur as monomers, chains of monomers, and branch structures. That would be the most accurate description for the structure of carbohydrates. Okay. Next one. Okay, so lipids now. Lipids, the pattern matching activity talked about lipids a lot. So I would actually say whatever's on the pattern matching is pretty much all you need to know about lipids. Okay, so the section on lipids for the pattern matching, that's what you need to know. As a recap though, lipids are not technically a polymer, but we say, we call it a macromolecule. And the monomer is fatty acid chains. Steroids are a type of lipid, and these are the ones that look like 4 rings fused together. Notice though that all of these rings, since they don't have any atoms around it, they're all just carbons and hydrogens. The structural features of lipids are predominantly CHO, okay, so C, H, and O, but mostly carbons and hydrogens. Because it is mostly carbons and hydrogens, that means that they are non-polar. Like, if anything, you should know lipids are pretty much, we're going to say they're non-polar. They do not like to mix with water. They are hydrophobic. Okay? All right. So when you are distinguishing between lipids and carbohydrates, one thing to note, they both have CHO as the elements, but for lipids, they're going to be mostly carbons and hydrogens. Whereas carbohydrates, it's the ratio will be carbon CH2O. The specific type of lipid that the book goes into is a triglyceride. A triglyceride is a type of lipid used for long-term energy storage in the body. It's made of a glycerol backbone with 3 fatty acid chains attached. So I included this picture just so that you could see this is a glycerol molecule. All right, so that would be what's represented by this head. And then the fatty acid chains, you saw that before. There are 2 types. We've got saturated fatty acids and unsaturated. The difference between saturated and unsaturated is that unsaturated fatty acids have a double bond. Okay, so that would be the key difference. Because they have a double bond, they have fewer hydrogens, because instead of having, this carbon has 3 hydrogens attached, if it's a double bond to another carbon, then it only has 2 hydrogens attached. Okay, now physiologically, this makes a difference in the foods we eat. So saturated versus unsaturated. Saturated fatty acids are solid at room temperature. We've covered this before. And if you look at the chain, they look straight. Unsaturated fatty acids, because of these double bonds, the double bonds causes a little bend in the fatty acid chain. So you get a little curved chain versus one that's completely straight. This curvature, this makes these unsaturated fatty acids liquid at room temperature versus saturated fatty acids are solid. So butter versus oil, that would be the 2 most common examples. So luckily, you guys, you don't have to memorize this, but it will save you a lot of time if you know it. All right. So, or, over the weekend, organize your notes as some okay, saturated, I'm going to write down. Saturated is, single bonds and solid. Okay. Right here, the reason why saturated fatty acids are solid is because since the fatty acid chains are all straight, they can pack closely together. Whereas unsaturated fatty acids, since they have these little bends in the fatty acid chain, it means that they can't pack closely together. And so therefore, I guess the way I would say it is they're kind of loosely packed. They can move around easier. So therefore they're liquid. Okay, I'm gonna skip this. The other last type of lipid is phospholipid, which also your pattern matching goes into. So the key idea for phospholipids is it is both hydrophilic and hydrophobic. This is the only type of lipid that's going to contain nitrogen and phosphorus, because in the phosphate head, there's a phosphate group here, and there's a nitrogen right here. So this is the only exception to the elements. The phospholipid is what makes up the cellular membrane, and they are arranged in a way where the heads are facing outside and then the tails are facing inside, because the tails block out the water. They do not want to be around water. So this right here, I would, if you need to, re-study the section in the textbook that talks about the hydrophilic head. So this would be polar, and the hydrophobic tail, which would be nonpolar. This term right here, I know it's really small on your screen, but later on, if you rewatch this, it's a phospholipid bilayer. Bi means two, so 2 layers. Okay. All right, I'm gonna skip those. I'll post them later. I'm gonna skip these questions to save time. Next one is protein. So this one I do wanna spend more time on, because for the pattern matching, they only talked about amino acids, and I'm gonna focus on building on the amino acids. So proteins are also known as polypeptides. There are 20 amino acids. So the monomer is amino acids. There's 20 different kinds. This is the general structure of the amino acid. One thing I mentioned before, but I'm gonna say it again, is remember the amino group right here. It can be written as NH2, or it can be charged, and we would just write this as NH3+. Same thing though, they're both considered amino groups. Carboxyl groups, same thing here, where it could be COOH, or it could be COO-, but they are interchangeable. The R group is the variable group, and we also call it the side chain. Okay. Based off your warm-up question today, I think a lot of you are familiar with identifying the R group. So here are some examples. The red sections would be the R group, and then the structural features of amino acids will be chons. So sulfur is included, and then they have the same common structure except for the R group, also known as the side chain. Now, this is the part where it might, if we were at school, I would practice a little bit more with you, but since we were out of time, we didn't get to practice as much. So I hope you're able to identify functional groups as polar or nonpolar. Remember, the only one that's nonpolar is methyl. This is related to page 59 in your textbook, where it lists all the amino acids and whether they're polar and nonpolar. For your pattern matching, this, I'm gonna show you just how I sorted it here. So on the left side is polar. I'm gonna show you for card 9, NH2, that's an amino group, so that's gonna be polar, that's why it's polar. Hydroxyl here, carboxyl. So I'm circling all the sections that would make the R group polar. Sulfhydryl, amino again, carboxyl, and then amino. So in order for you to determine whether it's polar, it should contain one of these functional groups. And then on the other side, this left side, these are all non-polar because their R groups are just hydrocarbons. So just C's and H's. So all C's and H's. This one has an amino right here, but overall, if you look at how many other hydrocarbons there are, it will still be considered more non-polar. This one has a sulfur, but it's not at the end. So this final methyl group would give it more hydrophobic properties. And then here we just got one simple hydrogen, and then we've got hydrocarbons here. Okay, so this would be how we sorted the non-polar versus polar amino acids. Okay. So these amino acids, now the pattern matching does not go into the actual protein. So I want to spend the next few slides focusing on actual proteins. We go from individual amino acids to a protein. This is the COVID spike protein. So it's part of the coronavirus, the outside. They have these little spike proteins. What they're trying to do is the vaccines that they're developing, they're trying to basically block these spike proteins. So this is a 3D model of what a protein would look like. Now, polar amino acids will be more on the exterior or exposed to the aqueous environment. Aqueous means like water. So if you look at a protein, the polar amino acids will be more towards the outside, the parts that will be actually touching water. The non-polar amino acids generally will be found more on the inside, so the interior, because they're hiding from the water. Okay, so I know for the pattern matching, like what's the point of identifying polar versus non-polar? It will go into like the larger protein structure. So polar is more towards the outside, whereas non-polar amino acids are more towards the inside. So how do we go from this protein, like how do we get this protein, 3D protein? And it's going to be based off the amino acid sequence. So if you look at this diagram of the amino acid sequence, all these 3-letter words like SIR, VAL, SIR is just the abbreviation for each amino acid. And they are all linked together, kind of like a long chain. So remember, polymers is a long chain of monomers. We'll generally also like number them off. And this will come into play more when we talk about DNA and how DNA codes for the amino acid sequence. So this amino acid sequence, though, is going to determine this 3D shape of the protein. Okay, now this is the probably newer thing that we haven't covered, is the 4 levels of protein structure. If you have your textbook handy, I would open up to page 62 to 63, put a Post-it on it. I would really study these 2 pages because I'm going to briefly cover it, but like you might want to reread this. So the 4 levels of protein structure, they're called primary, secondary, tertiary, and quaternary. So just going from small to big. I'll go through each one briefly and highlight some key ideas for this. Okay, this image, you can, when you look back at this, these slides, you can read this a little bit more carefully. This next one also, I put a second image for you to review. These 2 are good, diagrams that summarize this big figure 3.22. All right, so starting with primary structure. Primary structure, if you look at this diagram, you have pictures of each amino acid, and then the green part is the R group. So if you notice, the green part is the part that's different for each amino acid. They list each amino acid in 3 letters as an abbreviation. And then here's another way that is drawn just in this picture. So you have the primary structure, it's just each little circle is a different amino acid. The definition of primary structure is the sequence of amino acids in a protein. All right, it's just the order of amino acids. Amino acids are connected by covalent peptide bonds. So I'm going to highlight this right here. These covalent peptide bonds are each of these bonds in between the amino acids. It's a solid line, so covalent, meaning they're sharing electrons. And we also, in the textbook and on your exam, if they mention the backbone is this pattern of NCC, 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 which is nitrogen, carbon, carbon, nitrogen, carbon, carbon. So I'm going to show you on the diagram what I mean by that. But the backbone is this grayish area. Okay, so this gray section, if you notice, you have nitrogen, carbon, carbon. The next one is same order, nitrogen, carbon, carbon. Nitrogen, carbon, carbon. So each amino acid, it gets attached in that same order. So it's always gonna be NCC. The next one comes in, NCC. So in the next few slides, if we talk about the backbone, we're talking about this gray section. Now, the last thing that I want to point out for primary structure is these 2 terms. So I want you to make sure you write this down. Okay, so we've got the N terminus, and the reason why they call it the N terminus is because you have the amino group here. And then if you go all the way down, keep going to the very end of the protein, then you have the C terminus. The C terminus, it will stand for the carboxyl group. It got cut off here, but basically at the very end, you're going to have a final carboxyl group. So for amino acids, there's directionality. They go in order. So if you look at this diagram right here, this would be the start. I'll type this out actually. So this would be the beginning, which is the N terminus. And then if you continue all the way to the end, this would be the final amino acid will be the end, which is the C terminus. So for the amino acid sequence, there'll be a start and an end. All right, moving on. So this is primary structure. Secondary structure. Now, for secondary structure, there's two types. Secondary structure, you have something called a alpha helix. So this funny looking A, we pronounce an alpha helix. And the word helix is kind of like twisted or coiled up. If you remember, well, we had been learning about DNA, but DNA is described as a double helix, so two curved strands. There's another type called a beta pleated sheet. So this letter is beta. And then the beta pleated sheet is, it kind of just folds up. Kind of like, when, I don't know if people still do this, but when you're really hot, when you're younger, you take a piece of paper and you fold it back and forth to make a fan. That's what the term pleated means. I also think about the word like pleated, like a pleated skirt, kind of like a schoolgirl skirt, where it's like folded. So the beta pleated sheet is like folding back and forth. So there's two types of secondary structure. It says local folding creates the alpha helices or beta pleated sheets. So those are the two examples here. And the way this happens is because it's held together by hydrogen bonds among the backbones. So the previous slide, when I talked about backbones, NCC, this is where it shows up again. The hydrogen bonds, if you look at a drawing, hydrogen bonds are depicted by dotted lines. Okay, so they're not solid lines. Solid lines are covalent. Dotted lines are hydrogen bonds. Now, when I said among the backbones, if you look here, you can see this pattern NCC. You see how it just keeps going NCC, NCC. So this is a backbone that I'm kind of like highlighting. This is one strand, and then this is another strand, NCC. And the hydrogen bonds form between a hydrogen of one and an oxygen of another. Because remember, when we think about water, hydrogen is slightly positive, oxygen is slightly negative, because oxygen is electronegative. So the electrons get pulled closer to oxygen, which gives it a slightly negative, partial negative charge. And then that attraction is a hydrogen bond. On this example for the alpha helix, same thing. You've got a hydrogen from one backbone attracted to an oxygen from another, and a hydrogen bond forms. Okay. One thing when you're writing down the definition of this is for secondary structure, it's local folding. So within a protein, these interactions are in a small area of the protein. Okay. And the reason why I said highlight local folding is because for tertiary structure, the next one up, so level 3 is whole molecule folding. And it's the overall 3D shape. So if you look at this diagram right here, you can see there's alpha helices, and then there's also beta pleated sheets. So they tried to highlight in blue. But the whole molecule folding is like the entire thing coming together, the 3D shape. It occurs through interactions between distant R groups or side chains. So in this case right here, distant R groups is you have one amino acid, in this area and another amino acid in another area, but there are R groups can interact with each other, which causes it to fold up. This is in your textbook. So if I go too quickly here, please reread this section. Ooh, airplane, it's loud. But with whole molecule folding, there are 4 examples of these interactions that can happen. So the first one is, actually, well, let's just start. Let me give you some background on what we're looking at. So first, this purple line right here, this is your polypeptide backbone. So if I was to draw out the entire thing, it would be N, C, C, N, C, C, N, C, C. Okay, so this is the backbone. And then what you see coming out of the purple line, these are all R groups. Okay, so this entire thing that I'm circling is the R group. Actually, let's take a look. Let's see if we could figure out. Everyone turn to page 59 in your textbook. And I want to see if you could figure out what amino acid is this right here. So the one that's circled. So open up to your amino acid page. Once again, it's page 59. So page 59 in the textbook. And this is the R group that I circled. So look, scan through the images and see what amino acid is this that I circled. All right, I got some responses. If you didn't get a chance to look, take a look at serine. You are correct. So if you did not get a chance to look yet, look at find serine. So you would find serine on your amino acid table, and you can see that this is the R group, so CH2 and a hydroxyl OH. Now, how about, okay, so let me actually, so this is serine. How about this one below? See if we could figure out what amino acid this one is. I'm gonna look also because I don't know. See? All right, so look up this. All right, asparagine. Close. You're close, but it's actually, there's one. If you look at carefully at asparagine, asparagine has nitrogen and there's no nitrogen. All right, yeah, Kiara. Okay, so if you're still looking, Kiara did find it. It is aspartic acid. It's on the very bottom of the table. And so this second one is aspartic acid. Now, obviously, you don't have to memorize any amino acids, but what I did want to point out is based off of this diagram, you can actually figure out what amino acids each one is based off the R group. Now, for this, serine and aspartic acid, because of their structure, they are able to form a hydrogen bond right here. So this dotted line is a hydrogen bond. So the hydrogen of serine will be attracted to the oxygen of aspartic acid, and you can create a hydrogen bond. Okay, so that's the first type of interaction, is a hydrogen bond. 2 different amino acids, they attract each other, and you can form an H bond. Another type of attraction would be a hydrophobic interaction. So the definition of this is clustering of hydrophobic groups away from water and also van der Waals interactions. So if you notice these two amino acids, so I'm not going to spend time looking it up, but this is one amino acid. This is another amino acid. Okay, so, but they're both non-polar. Okay, so, whoops, that's really ugly. So this is non-polar. This one is also non-polar. Remember, non-polar things want to be around other non-polar things, so they attract each other and they huddle up close. Okay, so this would be another attraction that could occur. A disulfide bridge. This now is one amino acid. This is another one. These sulfurs can bind together, and this is called a disulfide bridge. And the last one is an ionic bond. So in this case, this is one amino acid right here. This is another amino acid. This one has a positive charge. This one has a negative charge. And obviously an ionic bond forms when the positive charge attracts a negative charge. So this would be the fourth type of interaction. I just really quick, so because I know if you watch this later on, you can take a look at this closely, but 1, 2, 3, 4, 1, 2. This would be lysine. And this one right here, oh yeah, thank you, Kiara. This one, whew, my gosh, this one is CH2CO. I think this is aspartic acid. All right, so this lysine is a base. Aspartic acid is obviously an acid, and the acids, since bases are positively charged, acids are negatively charged, they can form a bond. Okay, so for this slide though, key ideas is tertiary structure. These are the 4 common interactions that can form between R groups, and that holds together the entire 3D structure of the protein. Last one is quaternary structure. The quaternary structure, the definition of this is it's the end product protein, because many proteins are made of more than one polypeptide. So if you look at these diagrams, this is hemoglobin, and hemoglobin, they have 2 different types of chains. They have a beta chain. So there's 2 beta chains. These 2 are beta. And then there's also alpha chains, and these 2 are alpha chains. So each one of these, once they fold into the tertiary structure, they interact with other polypeptides to make the final actual protein. This is another example, collagen. In this case, you have 3 different polypeptide chains all interacting with each other. Okay, so quaternary structure is like the final protein. And a lot of times it needs multiple polypeptides to make your final protein. Okay, so that was a summary of the 4 levels of protein structure. Okay, it is 2:15 right now. My last one is nucleic acids. So if you want to stay to listen to that, you can stay. If you need to go, you can go, but please watch the recording of this later. Nucleic acids is the last one. All right. So for nucleic acids, objectives, once again, review that on your own. Nucleic acids are DNA and RNA. The monomer is a nucleotide, and there are 5 nucleotides. The general structure, you need to know this. So the 3 general parts of a nucleotide is you have the phosphate group, you've got a 5-carbon sugar, and then you have a nitrogenous base. Now, when you're doing the pattern matching, this looks very similar to sugars because they're ring-shaped, but the key is these rings have nitrogens in it. Okay, so that would be the way you would tell between a carbohydrate ring versus a nitrogenous base ring, is because obviously the name nitrogenous base, it has nitrogens. Here are some other examples, and on the pattern matching, the last 2 slides they mentioned, it could either be 1 phosphate group, 2 phosphate groups, or 3 phosphate groups. Structural features, so CHOMP, you have phosphorus with this. Rings containing nitrogen, the green, attached to a sugar, which is this reddish color, and then you have a phosphate group, which can range from 1 to 3 phosphates. For some other terminology, So this right here, you've got first the nitrogen base. Okay, so this represents the nitrogen base. There are 5 nitrogen bases, and they are defined as either purine or pyrimidine, 2 rings or 3 rings. So one thing I read was purine, the word is shorter, but it's a bigger ring. Pyrimidine is a longer word, but it's just a single ring. So you have these 5 choices. You have A, T, G, C, and U, uracil. If you combine the nitrogenous base with a sugar, there's 2 types of sugar. You got deoxyribose and ribose. The only difference between these 2 is deoxyribose has only a hydrogen here, whereas ribose has an extra oxygen. And notice they both end in ose, which means it's a sugar. Okay, so remember all these words that end in ose are sugars. This a nitrogenous base plus the sugar is a nucleoside. And then if you add the phosphate, then it's a nucleotide. So the terminology nucleoside versus nucleotide is nucleoside does not have the phosphate groups, but the nucleotide would. All right, so that's just like building from, piece by piece. I'll admit this person. Oops, okay. All right, so this is general structure of a nucleotide. DNA versus RNA, you should also know the differences between DNA and RNA. So 2 things that we've already covered. One of them is we've got in DNA, it's a deoxyribose sugar versus RNA is a ribose sugar. DNA, if you look at the structure, it's double-stranded. So 2 strands versus RNA is usually single-stranded. And then the last difference is the bases. So both of them contain C, G, and A, but one difference is deoxyribose has thymine, T's, and RNA is U. All right, so those are the 3 main differences: double strand versus single strand, the sugar, deoxyribose versus ribose, and the last one is DNA has T, thymine, whereas in RNA it's U, uracil. Okay, hang in there. We're almost done. Thank you for staying with me. Okay, so for base pairing rules, this is also going back to first year bio. For DNA, every time you see an adenine A, it's going to get paired with a thymine T. So if you look at this diagram, all the greens are represented so the greens are A's, purples are T's, and you'll always see that they're going to match up. Guanine and cytosine are also going to always pair up. So if you see a guanine on one strand, the other strand is going to be cytosine. All right, so that's always, we call those the base pairing rules. So the base pairing rules from DNA to DNA is C and G always match up, and A and T always match up. Now, later on, we'll talk about DNA to RNA. And so the one difference between DNA and RNA is RNA doesn't have T's. So every time, instead of adenine pairing up with a thymine, it would just pair up with a U, uracil. Okay. If you had me for 9th grade bio, I always would say, remember it as GCAT. Okay, you guys are gonna eye roll me, and I'm embarrassed that this is recorded, but GCAT. Okay, I always say, remember the bases, GCAT, because Jesus already showed you. Okay, G and C pair up, and then A and T pair up. So I remember it as in order GCAT, because G and C are next to each other, A and T are next to each other. So therefore, to remember this, it's a gangster cat. Like, oh my gosh, I cracked myself up. Just kidding. Okay. All right. But so G and C, they're next to each other. A and T, they're next to each other. All right. Now for RNA, I say it as GCAP, because instead of a T, we have a U, a uracil. So G and C pair up, A and U pair up, therefore, here's my final embarrassing picture for today. So hopefully that will help you remember the base pairing rules. Okay, so GCat for DNA, GCow for RNA. No, I didn't draw it. I actually Googled it, and I spent like 10 minutes trying to find the ideal pictures for this slide. All right, so but point is though, these are the base pairing rules. You should know that, write it down somewhere. Okay, so thankfully, you know, you don't have to memorize anything, you can write it all down, but it's always better to know some of these to save time on your test. I couldn't find a better one. No, not any that was like not offensive. All right, okay, so the directionality, proteins have directionality. I talked about this earlier. So we've got the N terminus and the C terminus. All right, so proteins have directionality, and now DNA and RNA also has directionality. So nucleic acids also have directionality, as in they have a 5' end, we call it a 5' end, and a 3' end. Since DNA is double-stranded, the other strand also is gonna have a 5' end and a 3' end. And just so that you guys know this, so I say 3, and this is like an apostrophe, but we call it prime. And same like in math class. So 5' and 3'. So when you think about DNA and RNA, I always want you to pay attention to these ends. So there is directionality. There's an order, like this is the beginning, this is the end. Similar to the proteins. This is the beginning, the N terminus, this is the end, the C terminus. So for this, I'm gonna show you really quick, it's like 1-minute video to summarize 5', 3'. I'm gonna narrate. I think it's just slides. So in the double helix form, when it untwists or unfolds, you can actually label the ends. All right, so double-stranded, double helix. Okay. All right, so I'm gonna pause right here real quick. So one thing to note is it's always gonna be opposites for each strand. So if there's a 3' on one, the other strand, that side is gonna be a 5'. It is related to Okazaki fragment, yeah, when we talk about DNA replication. For now though, all you need to know is that if you have 3' on one side, the other side is gonna be 5'. And so if this is 3' on the red strand, the other, if you follow it down, it'll be 5' at the other end. Now, whoops, oh crap. Now the way that you actually figure out what's the 3' and what's the 5' is based off of how you number the carbons. So this was when I was talking about glucose and how we number the carbons. A single nucleotide, you've got your base, you've got the sugar, which in this case, they're gonna use deoxyribose, and then you have the phosphate group. If you focus on the sugar, we number the carbons. So remember, we go loop around clockwise, and we come up with the 3' and 5' numbers based off of the number of carbons. So I'm gonna pause it real quick. So this right here, you see this carbon is numbered 3, this one is numbered 5. So if it turns to make your DNA strand, oh my gosh, why isn't it playing? The 5' end will be facing the phosphate. The 3' end is gonna be the other direction. This also, like what Chiara was saying, is when we do DNA replication, you always have to follow the direction of 5' to 3'. Okay, but that's getting ahead of ourselves. For now, the key idea is that the way we number the 5' and 3' is based off of the sugar. So this right here, this 3' carbon, this would be your 3' end, whereas this 5' carbon, so remember this little section right here, there's no carbon there, but there actually is a carbon, so this would be 5'. To summarize, DNA strands are complementary and anti-parallel. So these two terms will come up a lot. Complementary, the English definition of this, I copied and pasted it up here, is something which completes something else. So people would say, like, me and my husband, like, I'm really loud, I talk a lot. My husband doesn't. He's like very quiet. So people would say, oh, you complement each other well, because not that opposites are always, you know, what it should be, but, you know, it's good to have someone who listens versus someone who talks like me. So the term complementary is like, you know, it completes the other. So when we think about DNA strands are complementary, this refers to the base pairing rules. So each strand complements the other. So GCAT, our base pairing rules. Now the other word, anti-parallel refers to the strands running in opposite directions. So this goes back to the 3' and the 5' ends. So if one DNA strand, 5' end to 3' end, the arrow goes in one direction. For the other strand right here, you've got the arrow going up. Whatever is 5' on one end, they're like diagonal from each other. So like this. And then 3' would be the opposite. So that's the term anti-parallel refers to these ends. All right, I think that might be the last slide. Yes, that's the last slide. Okay, thank you. You made it. You made it. Thank you for sticking with it. Now, there are practice questions on here, but I skipped them to save time. Before I end this, a couple last things. Let me just show you on the slides for you to review on your own. So after this, so when you open this up later, if you continue on, I actually included a few slides for chapter 2 and 3 review, because it's not just macromolecules. We also covered chapter 2 and water. So chapter 2, 2.1 to 2.4, I call this chapter 2A. This was the basic chemistry, so reviewing bonding, polar, nonpolar, and hydrogen bonds. Chapter 2.5, this is a big one. So this was properties of water. All right, so I included some slides, the key slides for these topics. And then 3.1 was carbon and the functional groups, which we talked about today, but just wanted to remind you though that you should still study water, properties of water. Okay, so on these slides right here, if you continue down, like I have a few slides I picked out that I thought were the most important. So you can scroll down and study these on your own, including the objectives. So I also have objectives for each chapter, each section. You also are welcome to obviously look back at our old slides. I would say for the weekend, one way to prepare is read over everything for your notes, your study guides, go through, highlight things. If you want to compile everything in one area so you could refer to it, do that. Yeah, there's some encouragement from Mr. Martinez there for you. But yeah, thank you for sticking with me. I know it was a really long lecture, and if you have questions, you can message us. But make sure, I'm gonna go back to this, my first slide. I don't even know where to start. It's so long. Make sure you guys do the Unit 1 practice FRQ. Oh, you know what? I skipped some. So, okay, sorry I keep talking, but on AP Classroom, the progress check is due. So you have to do that one. Now there's also a Unit 1 guide, and this is actually what AP teachers use to prepare our lessons. So this one, it says Unit 1 Chemistry of Life. This is, if you hear about standards, like your teachers have standards that we have to teach, this is it. Okay, so if you want to know exactly what would be on the test and what's on the progress check, it's this file right here. It looks really intimidating, but it just summarizes everything that you need to know. So this could be another option for you. They break it down by topic. So the first topic is structure of water and hydrogen bonding. The part that you need to look at is the essential knowledge. So this blue section is like what you need to know. So if you look at, if you want to look at that could be helpful. I didn't realize I skipped a slide. The other thing that is helpful is AP Classroom also has daily videos for each topic. So if you want to watch these, you can watch these. I already mentioned it, the study guide in your notes, Bozeman videos, Khan Academy. There's a whole unit that they have on what we were studying. So I put the link here. And last one is crash course videos. I like this guy, but he talks really fast. So sometimes it's like he goes so fast I can't follow. But now that you've at least covered everything once, it might be useful to just review. So, okay, these are all options for you, the study tips. Okay, I am done. I'm gonna stop recording. Um, let me, that'd be funny if this whole time I did not record.